All right, so my name is Luis Helfer, and I work in the Shorts lab, and we work with a special class of proteins that we call the FET class of proteins. So before we start, let's first talk a little bit about what a protein is. So a protein is basically just the workhorse of the cell or of the body. So in this case, there's three proteins that we study. One that's called FUS, another that's called EWSR1, and the final one that's called TAF15. And together, that's why we call them the FET protein. So they have mainly three separate domains. The first one is this LC domain here, which is just a low complexity domain. And this is where most of the assembly into fibers comes into play, and it binds to RNA Pol2 in this case. The final two domains, this RNA recognition motif, which is the RRM, specifically binds RNA. And then the last domain has three separate subsections. So we have this RGG2 and RGG3. And these are just arginine and glycine rich domains, which are amino acids. And arginine, we know, has a positive charge on it. So it's really attracted to the negatively charged backbone of DNA and RNA. So that's why it'll bind RNA and assemble into fibers in this case. And then so you can see that each FUS, EWSR1, and TAF15 all have the same domain sections, but they're different lengths. And that's because they specifically have different roles within the mammalian cells. All right. So in this case, you can see how these fibers begin to form. So we have this RNA sequence here, and this tail is the low complexity domain, and then this sort of head is the RNA binding domain, so the second two domains. And those will sort of seed onto an RNA sequence and then form higher order assemblies through interactions with this low complexity tail between each of the separate proteins. And then RNA Pol2 can come in and directly interact with the low complexity domain, or sort of come in the middle of it. And it's basically just two separate interactions between the two. And then so the one I was studying specifically was EWSR1. So EWSR1, when it becomes mutated, causes a specific kind of cancer called Ewing sarcoma. And the sarcoma is just a tumor found in the bones and the soft tissues. And in this case, it is the second most common form of pediatric bone cancer. And it's very aggressive. It has a 20% survivability rate once it's metastasized. So it's pretty aggressive. And then so what happens in Ewing sarcoma is we have this healthy chromosome 22, which is EWSR1. It has all the domains, has the LC domain, RGG, the RNA recognition motif, and the final portion of it. But what happens in Ewing sarcoma is that in chromosome 11, we have FLY1, which has two separate domains. It has an activation domain and a DNA binding domain. And what happens is this DNA binding domain will translocate with this domain, and it'll connect to the LC domain of EWS. So this forms a mutant called EWS FLY1. And what happens is that the DNA binding domain sort of allows the protein to keep binding DNA and activate transcription. And what happens with that is we know that it'll form a whole bunch more proteins, and it'll lead to rapid cell division, which will lead to the tumors. So that's kind of what I was already saying. So this DNA binding domain translocates with the LC domain. And then it forms EWS-FLY1, which is this final product. So what my project was, specifically what we were working with the EWS-FLY1 and EWS-R1. But at the time, they had a kind of buggy tag, which a tag is just so we can isolate it after we grow them up in E. coli cells. So what happened was they had a glutathione S transferase tag, which is just GST. And we had to switch that out with another tag that we knew would help isolate the protein even better and give us higher concentrations of the protein. So what I did was I had to switch the tags, switch them out. So basically what we have is we have this EWSR1 gene, and then we have this other plasmid with this MBP tag. And what I wanted to do was insert this MBP tag in front of this EWSR1 gene. So in order to do that, I had a couple options. I could have used restriction enzymes. I could have done a couple of different PCR methods, but we settled on this specific method called quick step cloning. So instead of using the typical restriction enzymes, which give you blunt ends within the DNA, we used this sort of novel idea where you order this primer with this white portion, which is where you're going to insert it into the new DNA, and this blue portion, which is the old portion of the DNA. And what happens is DNA polymerase will sort of act, if you don't know what DNA polymerase is, it's like a copy machine. And it'll jump on and copy it and form these separate strands of DNA here. And then once they begin to anneal, they will form these asymmetric primers. And they're asymmetric because they have sticky ends on each side. And these sticky ends will allow the DNA to sort of bind to the plasmid of interest that we want to insert it into. And then the DNA polymerase can just jump on right here at the end and sort of copy all the way around. And then you get the final product. It's pretty simple in like context, but when you're doing it, it can be a pain. So what I did was I kind of showed just a quick snippet of like what the data looks like. So this is the asymmetric PCR product. So it would be this section right here. And what we have is just the size of the DNA, which shows us that it's at the right size. And then the concentrations of DNA that we got. And then from there, you just, in hindsight, you just basically mix it back in, set the program, and then it'll give you this. And then you put it into bacteria. That's the gist of it. And then that's my project so far. And we're working on what to do next. So thank you.